And for those who enjoy this channel and would love to support us financially, please feel free to hit that donate link. We'd greatly appreciate it. God bless.
Okay, we are live on Standing for Truth tonight. We've got a really good stream. We got some good feedback over our stream the other night on distant starlight. We're going to be doing various episodes, you can call it, on the so-called best evidence for uniformitarianism and universal common descent. So today we're going to be going over nested hierarchies. So a lot of this is going to be technical. We're going to try and give visuals along the way as well and screen share some supporting papers. But I've got Jeremy here as well, who's a genius on this topic. This guy has taught me a ton. As I said it before, he's been a tremendous blessing to this ministry and to the work that we're doing. So God bless you for that, Jeremy. Thanks for joining us here tonight, Jeremy. Did you have a few words at all to the audience, brother?
Hello, and thank you for the, correct me if I'm wrong, the English compliment, standing for truth, my friend.
No problem, man. It's easy to compliment you, brother. You've taught us all a ton. And like I said, you've been a tremendous blessing to this fight. So we're always happy to learn from you. And I think this is one of Jeremy and I, both of our favorite topics. So we're going to have fun tonight. And I want to point out to the audience as well, if you have any specific questions, tag Praise or myself, and we'll make sure that we answer those throughout the stream. So let's get this going real quick. I do have some visuals. I'm just going to, like I said, a lot of it's going to be technical. I'm going to start off with kind of a simple explanation as to what the evolutionist means by nested hierarchies, and then kind of what we mean by it. Then we're going to have Jeremy jump in here, and we're going to get a little bit more technical. We're going to refute Herman May's. He looks to specific gene trees, like neutral variation in amino acid trees, for example, third position codon variation. Conspiracy Cats, who I've debated, him and Dr. Stefan Frello look to nested hierarchies in mitochondrial DNA proteins, specifically the cytochrome C. So we're going to go over that and refute those as well. So these are typically the best lines of evidence that the evolutionists have at their disposal, and I guess I'd have to say your best evolutionists utilize them. Herman May's, Conspiracy Cats, Aaron Ra more so in anatomy and morphology and physiology. So we'll address that. But genetics is kind of where the war is won. Genes and traits, that's what's inherited, sperm and egg. So that's what we're primarily going to focus on today. Yeah, so let's get started. I'm going to show just a visual of what we mean by the nested hierarchy. So I'm going to share screen right now real quick. It's just going to be on a slide. And okay, let's see here. So as long as we can see, Jeremy and Brother Praise, you can see that?
It's a little small.
Okay, that's fine. And Praise may be able to zoom in as well. So let's just go over the hierarchies in general and the parts of it that are agnostic, for example.
Before you get going here, SMT, do you see that little thing that there says hide? It just makes it so much easier. It makes more room.
Do you want me to click hide?
Yes. Boom! Nice. Yeah.
And then how do I, when I want to stop sharing screen, how do I do it?
I think it'll show, but if you just put out your just hit stop sharing at the bottom.
Oh, you mean I gotta go once I go back into the stream part?
Yeah, just hit the very bottom square. It'll say remove from stream or something like that. Where all our boxes are at the bottom.
Okay, yeah. So right now I'm in the actual slides. Once I go back into the Streamlabs, then I can click it there. Okay. So right off the bat, guys, you can tell that just if you just stand back and look at their hierarchies and their diagrams, it's not that impressive because obviously we're going to expect more similarities in, for example, humans here and chimps than we would with fish and a salamander. We're going to expect more similarities in anatomy, morphology, physiology, and genetics with the human and the chimp than with the salamander and the fish. And even if you just get onto, like, if we look at apes, for example, apes and humans, old world monkeys, down to the lemurs, same thing applies. We're going to expect more similarities in these things: anatomy, morphology, genetics, and physiology with humans and gorillas and chimpanzees than we will with, say, the New World monkeys and down to the lemurs. So if you just stand back and look at it, this is pretty obvious. But let's represent the evolutionists appropriately here. Here's a good diagram right here. Humans, primates, mammals, vertebrates, invertebrates. We can illustrate this nested hierarchy that the evolutionist talks about. Let's employ humans as a foundation, okay? So we all have enormous amounts of features in common between any two humans. And this observation places us, as you can see here, in the group humans. Now, how much do we have in common with a chimpanzee or an elephant, for example? Well, we as humans all share much more with each other than we do with a chimpanzee, of course. But we as humans actually have a lot more in common and share many more features with a chimpanzee than we do with an elephant or a horse. I think this is obvious. This is that groups within groups patterns that emerges when we begin to classify life. So to simplify things, we're all grouped as humans. We are also grouped together with primates, and then we are grouped in an even more inclusive group called mammals. Why? Well, me, Jeremy, Praise, for example, here have a lot more in common with chimpanzees, monkeys, elephants, and whales than we do with fish. But all of us collectively with the fish have more in common when we consider a backbone, for example, than we do with, say, an insect. An insect is an invertebrate, which lacks a backbone. So that being said, we are classified with the insects as animals, and so have more in common with each other than we do with plants. Once again, this is the groups within group pattern of life that the evolutionists say demonstrates descent with modification. The evolutionists will say, though, that these nested patterns can only be explained by common descent. Why do we have much less in common with a fish than we do with a chimpanzee? Well, the evolutionists will say this is simply a reflection of time, and we have a much more recent common ancestor with the chimpanzee than we do with a fish or an insect or a plant. Hope this is making sense, everybody. But the evolutionist says, this groups within groups pattern is expected if universal common ancestry were true. Well, it turns out, and you can see here on the design mode of transportations, that the means of transportation that humans design naturally fall within a groups within groups nested hierarchical pattern. We do this by accident. We don't even do it on purpose. That's what's funny. So all sedans are incredibly similar to each other. Sedans are much more similar to each other than they are to SUVs. Sedans and SUVs are more similar to each other than they are to tractors. Now, sedans, SUVs, and tractors are similar enough to each other that they can be grouped together as land vehicles, which would also now set them apart from, say, boats or airplanes. This is the same groups within groups nested hierarchical classification that emerges in design modes of transportation. Now, the evolutionists, when I'm debating them, will often try and point out some inconsistencies in the design modes of transportation. But guess what? The biological nested hierarchical patterns that you see, there's always examples you can point out that are contradictory. For example, anytime they invoke convergent evolution, they are admitting to some type of lack of uniqueness in the phylogenetic tree or incomplete lineage sorting as well. For example, you can take different genes and sequence those genes, and you will get different phylogenetic results, for example. You can even just see that in the great ape family when they try and classify humans with the other great apes. Also, there's something called loss of traits evolution. So there's inconsistencies in the phylogenetic tree of life, just like they may be able to point to an inconsistency or two in the modes of transportation, nested hierarchy. But the rule is hierarchies exist. So the fact of hierarchies and the point, I know this was long-winded, but it's important, and I think the visual is important. The fact of hierarchies is agnostic to the debate. That's why tonight Jeremy and I are going to focus on that which is which can differentiate between the models. Is it nested hierarchies by descent or nested hierarchies by design? And that's what we're really going to cover here. So, Jeremy, okay,
Yes.
so I just clicked on the screen here. Yeah,
Yes.
I know that was long-winded, but I just wanted to represent as best as I can what the evolutionist means by the nested hierarchy, and I think I did so well, and then kind of show why both models would expect the same thing. So that's where we're going to kind of segue into the lines of evidence that can differentiate between the models. So Jeremy, maybe you got a few things to add there as well. I may have missed a few things.
Yes, you did miss a few things. Sorry, you were saying?
No, I'm saying the floor is yours. Maybe you wanted to add a little bit to that before we move on to the evidence that differentiates between the models.
At the species level, both models do predict the same thing, but on the family level, well, both models do not predict the same thing. Okay, that's what you missed. That's one critical thing you missed. So you asked me what you missed, and that is what you missed. That's a critical thing. Both models do not predict the same thing overall. Okay, we predict this particular. Sorry for my English. I'm not good at it. Very good at it. I'm not an American or from the West. I'm from Asia. This animal would speciate, and there would be hierarchies. Okay, there would form a hierarchies as it speciates into different species. That is part of the created DNA differences that the creator put in animals, and those created differences. As you can see, there are many types of insects. There are many species. One insect has so many species, but still, it is the same insect. And there are several types of birds. Okay, so it means that the creator put more DNA differences in animals than humans, because as you can see, humans are so similar, right? So okay, so I'm going off the topic here, but my point here is both models do not predict the same thing. At the species level, yes, but not at the family level. Okay, we predict a cow would produce a different type of cow, but still a cow.
Well,
But
well, real
yes,
quick, I'm saying that we expect and predict. Well, oftentimes evolutionists will say, well, you didn't actually predict it. It's a retrodiction. Well, for example, Linnaeus, he invented the classification scheme, Linnaean
yeah,
taxonomy, for example, kingdom, phylum, class, order, family, genus, species. And he classified life based on function. So in a way, yeah, we did predict it, but a lot of it is retrodiction as well. But that's why the differentiating evidence would come down to predictions. But yeah, like you said, and I think that's a good point you made regarding the created heterozygosity hypothesis. So if God, as we propose, obviously he didn't mean for Adam and Eve to reproduce by cloning. That wouldn't make any sense at all. He said be fruitful and multiply. So if Adam and Eve were created with those pre-existing functional DNA differences, and that model, that hypothesis applies universally among all forms of life, all the different kinds that God created in the beginning, here's what the audience needs to understand. And this is kind of what Jeremy pointed out. Even if scientists showed up just an hour after the creation event, okay, and they started doing genomic sequencing, or they started analyzing morphology and anatomy, what they're going to find are nested hierarchical patterns that they can now conclude are the result of universal common descent, even though this is only an hour after the creation event. Those pre-existing functional DNA differences give us the expectation of nested hierarchical patterns. Now, as Jeremy's saying, since the creation event, there has been speciation events that have always reflected the initial hierarchical patterns, which means we will see hierarchies in the biological world, just like the evolutionists will. We're going to see more in common in anatomy, physiology, genetics, and more morphology, as I said, with the human and the chimps and the old world monkeys and the new world monkeys, than we're going to see between humans and fish, or humans and amphibians, humans and bacteria. So that's that nested hierarchical, that's that tree of life. So yeah, we both expect, even the bigger picture, Jeremy, we both expect the same thing in terms of these patterns.
Yes, we do expect the same thing, but not very specific. If my English is correct. I mean to say.
Are you saying you want to go into the different? Because I was showing the commonality first, just to represent their side, represent our side, showing that which is agnostic. Do you want to move on to now that which can differentiate between the two?
Gnostic. What is the meaning of Gnostic again?
I understand then the miscommunication. Agnostic means it's neutral. So the hierarchical patterns we see, and as I pointed out, the anatomy, morphology, physiology, and genetics between all forms of life is expected between both models. Therefore, if we're going to try and figure out what model is true and which one is false, now we need to look at lines of evidence that can differentiate between the two. Because so far we're discussing that which both models can explain. Now we need to look to that which both models can't explain. We got to rule out one of the models. So I think we both know how to answer that. Is that what you wanted to move on to then?
It's possible, but don't forget the fact that we can narrow it down into, for example, all birds, all types of birds, all families of birds can share the same hierarchical pattern. All flying creatures, no matter what family, which family, can share the same hierarchical patterns. Okay, and all land creatures can also share the same hierarchical patterns, and all sea creatures can also share the same hierarchical patterns.
Exactly.
Okay, so that is one way to narrow it out.
Well, because, for example, when Aaron Ra might say, like in his phylogeny challenge, where do we draw a line? Well, even though we would say not every single bird is related through common ancestry, that God created multiple kinds of birds. Well, even those different kinds of birds, like you're saying, all the different families of birds, they're still going to share nested hierarchical patterns. Even though, according to our model, not all of them are related. But according to the evolution model, they are all related. Therefore, those patterns reflect divergence time, speciation events, for example. But for us, there would be a line somewhere in, like you said, all the birds that would form a family, all the reptiles. So that's all
land creatures,
that are land dwelling. Even though we wouldn't say all animals that are land dwelling are related, but we would say, based on those pre-existing DNA differences, even at the moment of creation, you take all land dwelling creatures, even though God just created the different kinds, if you start sequencing their genes or looking at anatomy and morphology, you're going to find hierarchies. There's hierarchies everywhere.
Yes.
Right.
Hierarchies, which extend beyond one family to another.
Exactly. There's nowhere in the biological world where phylogeny breaks down.
Actually, I can break it down. If we go specific with the number of nucleotides, amino acids, and so on, we only look at
exactly, and that's where we now get into the differentiating evidence. I'm just saying the fact is nested hierarchies exist in all forms of life.
Yes.
Now, as long as we're through with that part, now we can get into where the hierarchies that are seen whether or not they break down, or what observation regarding hierarchies can show that the hierarchies are by design versus descent. That'll be the next phase. So if you want to lead into that phase, Jeremy, why don't you get us started as to what evidence, what we can look to demonstrate that these hierarchies are that of design.
Let's start with pseudogenes. Are you okay with that, standing for truth?
Yes, I'm going to mute myself. And you got the floor.
I'm sorry, you were saying?
Well, I'm just saying I'm going to mute myself, and I think that's a good place to start. So I'm going to give you the floor. I've talked a lot. I'm going to give you the floor to. That's a good place to start. Pseudogenes.
Yeah, pseudogenes nested hierarchy. Okay, I'll elaborate a bit or not. Okay, a bit, whatever. I did discuss a long time ago about. I'll give one example, because there are, like you said, standing for truth. There are hierarchies everywhere. Yes, I agree. I've studied it. Yes, and there is no better example than the olfactory genes. You guys know the olfactory, right? So those olfactory contain genes in which control the olfactory. Now I'll explain more. Okay, let's see where I should start. Okay, because I have memorized the numbers through the years. So yeah, sorry. By the way, another thing to call pseudogenes are pieces of non-functional DNA in a state of mutational drift. Ever heard that before? Mutational drift? Race or standing for truth?
Yes, we have.
Yeah, okay, thank you. Mutational drift, just like genetic drift, they are the same. In by principle, is principle the correct English? They drift, okay, forming the hierarchy. Okay, so it's called mutational drift. Just like the evolution of the English language, in which standing for truth is in Dr. Venom's book. So the English language has drifted. We can call that the English language drift, where in one generation the English language has a change. Okay, are you guys familiar with that? Yes, yeah, standing for truth is familiar with that. Okay, and then another generation, the English language has changed. Let's say the word Jesus has evolved into another word, which is also Jesus, but the spelling is different. So that is the mutational drift. A particular, let's say Jesus is a consists of several genes, and then that mutational drift would would uh, would evolve into another name, but it is still Jesus. Okay. Sorry. It will. The spelling would be different, but it is still the meaning of that of that is still Jesus, but the spelling has changed, and you can observe it in the evolution of the English language ever since the 1500s, 1400s, 15, 16, 17, 18, up to now. Okay, now, okay, sorry. I'm going somewhere else, but I have to explain the mutational drift first, so that the audience can understand another way to interpret the nested hierarchies. Okay, like I said, there's no other better examples that evolution is used, which is in Dr. Venom's book, which is in Dr. Francis Collins' book, and even in sorry, I really hate this guy, in Richard Dawkins' book. Okay, now the evolutionary olfactory receptor genes becoming pseudogenes is the result of the reduction of the importance of the olfactory in primate evolution. Okay, now standing for truth, you did talk about primates, correct? As the primary example.
I did. I had a diagram.
Yes, yes. Okay, now I'm gonna try to put in the numbers, okay, and make it more difficult for the evolutionist. Okay, now I'm not going to be specific on the numbers because I forgot the exact number. So I'll just say, let's say the number is exact number is let's say 185, 184. I'll just say somewhere around 180 like that. Okay, and that's enough for evolutionists to make calculations. But I will not drift apart so far from 180 to 20 or 10. So it's a more or less. I don't remember all the numbers. I did not check my notes from 7 years ago. Okay, so it's dusty. Now, okay, where was I? Yeah, the evolutionary receptor genes is one of their best examples. Okay, it is these pseudogenes was the result of the reduction of importance of the olfactory in primate evolution. This olfactory receptor that they used have actually collapsed. Many people don't know this, but evolutionists still use it. And bit by bit, this crap, this English, this pseudogenes here and there are collapsing at a rapid rate. Yeah, I already said it. Sorry, but evolutionists say they are just being turned off and happen many times in evolutionary history. So they have the luxury to say it turned on and it turned off. Okay, and we do observe it being turned off today. So they could infer that it also did turn off in the past and that it turned on right now. Okay, that is why they look functional. Okay, so that is their counter argument against your function standing for truth and Dr. Nathaniel Gibson's um, function issue. Okay, but I'm going to go deeper here. Okay, and by the way, a bit by bit collapse, okay, of this evidence is not what young Earth creationists need. Okay, the collapse has to be not only bit by bit, but the locations of the pseudogenes now with function has to be closer together and a lot more. They have to be closer. I mean, the pseudogenes here have to be closer to the other pseudogene here, which provide a a what you call that, an integrated functional pseudogene set. Ever heard of this standing for truth? The pseudogenes have to be closer together. Heard that? Because the pseudogenes that we observe are scattered everywhere. Although some pseudogenes have a specific function, evolutionists can say they were turned on and off. But if these pseudogenes are close together, then how can they say they were turned on and off? Okay, these pseudogenes would form a function in which sorry, I don't know how to say it in English. They would create a function that is more clear than the function that we find if those pseudogenes are scattered all around. And you agree that those pseudogenes are scattered around, correct? Standing for truth?
I would agree. I would agree.
Yes. Okay. All right. Okay. And a lot more. Okay. Are you familiar with wait, let me think again. Let me give an example. I am not okay. Let's say sorry for my English. Marmosets. I don't know how to pronounce it very well in English, but marmosets. Are you familiar with that? Marmosets. Marmosets. Okay, all right, never mind. Me asking you a question. Marmosets is a type of monkey, as I recall, which I studied a long time ago, with a sense of smell, because we're talking about olfactory, remember, okay? With a sense of smell that is strong and many olfactory receptor genes. Sorry, it is a type of monkey with a sense of smell that is strong and many olfactory receptors. It also has many olfactory receptor genes, pseudogenes. Sorry, not genes, pseudogenes. Okay, for us humans, for all their, for our, for their and our degenerate, their olfactory receptor pseudogenes, these marmosets, monkeys, their all olfactory receptor genes are degenerate, but still functional. Okay, so this is one thing that evolutionists are misleading people. Okay, when you say something is junk, then there is really no function. But when you say something is degenerate, it doesn't mean there is no function. Okay, right? Do you agree? Okay, so it is still functional. There is still the sense over there. The senses are still there. The olfactory sense is still there. Olfaction sorry. I'm trying to speak fast because I think standing for truth has many more to say. So my English is weak, so forgive me. I'm trying to make it as fast as possible. My grammar may have any mistakes. Sorry for saying that. Let's go back to topic. Um, we do observe olfactory receptor genes that have recently recovered, or in short, like I said before, turning on of the olfactory genes. So as you can see, there is function, correct? Right? Now, plus, like I said, when you say it's degenerate, then it is still functional. Degenerate, but still functional. Okay, if a car is rusty or is old, like Bumblebee's car in Transformers, in the new movie which features the girl, I got name. Oh no, in Transformers 3, sorry, where Witwicky used an old version of the car of a Bumblebee, but it's not Bumblebee, but it's just designed like Bumblebee. The car is really highly degenerate, but it's still functional. Okay, so a highly degenerate pseudogene does not mean it is really a pseudogene. That is a very misleading thing that evolutionists are pushing. Do you know what about this standing for truth?
Oh, sorry, I was on mute, brother. Yes, yes.
Okay, so degenerate does not mean no function, correct? You agree?
I
Okay.
agree. I agree. I think of it like well, I mean, that's going to be the next topic, the third position, a
Hold
codon
on.
variation that they often say is degenerate. Well, if
Yeah.
those third positions are functional, well, degenerate doesn't mean junk. So yeah, that's the way I see it.
Yes. I'm also going to talk about codons here as it relates to olfactory. Do you know there is a codon that's related to the olfactory? Okay, and there are other codons that are related to other
Well,
parts of the body.
before you do go to the codons, though, let me know because I want to illustrate Dr. Herman May's his argument on it, just to represent their side, and then
Yes.
we'll refute it. So just let me know right before you get to the codon part. Let me know, brother.
I'm almost there. I'm just trying to refresh my memory because I'm not able to find the paper in which I wrote, in which I studied about this olfactory the numbers. So I'll just give a more or less number. Okay, now the codons. Yeah, you talk about the primates. Okay, let's talk about the codons over there. Now for the primates, which is the big deal, right? That is the biggest deal, right? Sorry, biggest deal. That is the most important. Do you agree with that, right? Humans to primate evolution. That is the most common they use. Okay. Now for primates, let's say even if they are non-functional. Okay. All right. Those are olfactory and other parts of the primates, because, like I said, every part of our body has some kind of hierarchy. Hierarchies are everywhere. Okay, we have to be clear on that. But like I said before, the hierarchies has within the animal kind only. That it does not have to. It must not extend between the families. Okay. So certain primate pseudogenes contain what is that again? Contain a phylogenetic. I'll just remove the word philo. It contains a genetic lack of harmony. Okay, have you heard of this? It contains now for the primates compared to the humans. Let's say even if they're non-functional, certain parts of them, like the olfactory, certain primate pseudogenes contain a genetic the philo. Sorry, I'm not going to say philo anymore. Just genetic, because if you say philo, then some people might say it's about anatomy. Okay, so these pseudogenes contain a genetic lack of harmony. Okay, so because people are misled to believe that this hierarchy is perfect, but it is not. There are a lot of gaps in these hierarchies. I have seen them myself. The numbers do not fit, and there are a lot of gaps. There are, for example, the hierarchy is like this. There is just one number. Number one is over here. Number two, but there's no number three. There is no number four, number five, and the number five, for example, its function is different from number one. There are those examples. Okay, so I can name a lot of examples of that. Okay, but since they assume evolution, they form the hierarchy, and they predict that one in the middle of one and three is two. Okay, some did, some were able to successfully predict it, but most not. Yet they use it as evidence. Okay, and don't forget, standing for truth. You know that some no many of those hierarchies only contain a few nucleotides. Okay, only contain a few nucleotides. So it's like cheating. Okay, you do not form a hierarchy based on just a few nucleotides. You need more than that. Okay, right? The peptide gene is one example of that, and I can name a lot more. Okay, so going back, sorry, yeah, I'm not really going off topic here. So okay, so certain primate pseudogene contain a genetic lack of harmony. I gave the example. Okay, the example a while ago. Okay, the numbers. Okay, genetic. We are now assuming that there are the number of nucleotides are. Hang on, just 5 seconds. Okay, sorry about that. I just charged my. I'm just trying to charge my laptop here. Okay. There's a lack of harmony, and the example I made is the lack of harmony I was talking about. And there are a lot of examples of a lack of harmony in these nested hierarchies. Okay, it's not just like this. There are others. I'm talking about from one family to another, because this is the one that we have to debunk. We have no problem if it's at the species level that an animal kind would create a nested hierarchy, but contained within the animal kind. Okay, and one way to test it is breeding testing. Okay, so that would differentiate between the. That's one way to differentiate between a family from another family, right? A dog cannot breed with a cat. Okay, now going back to genetics. So I'm just trying to make it easier to understand, because this is highly technical and I do not remember all the data. Okay. Okay, the lack of harmony. I give the example. Okay, there should be a harmony where they, if the, where there should, if there is a, if the harmony is perfect, then they can perform their jobs effectively. And we do know that some genes do not perform their jobs effectively, right? Standing for truth. That is why some become junk, right? They degenerate, right? You there? Are we on the same page here?
That's right. That's right. They degenerate. They degenerate.
Yes, they degenerate because because there is a lack of harmony in the nested hierarchy that we're talking about. Okay, so there's a lack of harmony where there should. Okay, we're clear with that. If there is a lack of harmony, then Then why? Because they have degenerated. So number 2, for example, position number 2 has degenerated. Okay, so if they predict there is a number 2 in the middle of number 1 and 3, okay, this prediction will fail because it has already degenerated, and no turning on and off can happen because it is totally off already. So that means there is a lack of harmony over there. That is why we are degenerating. Like the genetic entropy you are pushing, my friend, standing for truth. Okay, now, and okay, so yeah, it is also a hint of function again. Okay, all right. If they are not functional for the primates, their genes that evolutionists claim are not functional. Certain primate pseudogenes should perform their jobs. Sorry, I'm repeating myself. Sorry, I don't remember all the data. It's premature. It's like a premature codon. Here we go. It is like a premature codon and a duplication. You agree with this, standing for truth?
I do.
Yes. Okay. An example now. I'll give an example. Let's say, let's move away from primates for now. For now, temporarily. Guinea pig. We know what a guinea pig is, right? Okay. There's a discovery of the fact that the independent is independent the correct English. Independent guinea pig and human gulo pseudogene. Here we go. Standing for truth. You are familiar with the gulo pseudogene, right? Now, the guinea pig has a discovery or this gulo pseudogene. The fact that a guinea pig and a human pseudogene have a 30 plus. Like I said, I forgot the exact number. I think it's 36% identical. Can you fill in, standing for truth? Am I correct with the number? Is it 36% or 30%? Or somewhere? Am I correct?
It's in that range. It's in that range. I'll have to look it up.
All right. They have 36. I remember it's 36. It was almost 6 years ago. I forgot the exact number. Sorry. So I'm getting deep into the numbers here to make it harder for the evolutionists. Now, they have a 36% identical unfitness. Okay? Identical unfitness. I think my English is going over. So they have a 36% identical unfitness. Do you know what I mean, standing for truth, when I say identical unfitness? I just want to make sure that my data is in sync with yours.
I do. I think you're explaining it very well, actually. Your English is good, brother. Keep going. You're doing good.
No, not my English. The identical unfitness. Heard of it? Never mind. Okay, now if this is true, this 36% identical unfitness, if this is true, wait, forgot again. If this is true, this helps disprove the entire evolutionary shared mistakes argument. I'm not kidding. Okay? Because this identicality must not be the 36%. They should be far apart between the humans, gulo, and the guinea pig. Okay? This argument, I suggest, do depend on the fact that pseudogenes are lacking in functions. Take note, lacking in function does not mean no function, like I said above. Like I said a while ago, sorry. But evolutionists say that they are junk. Now that's misleading. Sorry, I'm repeating myself. Now, there's the reuse thing about nested hierarchy. You heard about it, standard for truth? Never mind. God does reuse things, as you know, right? We got samples of that. Reuse, as in recycle, right? So the same,
That's right.
the same as pseudogenes. It gets recycled. If you heard of it, because God reuses pseudogenes. That's one reason they turn on and off. Okay, so as you can see, we have a counter explanation to Dr. Venema on the turning on and off of genes. Because why not God? Why can't God reuse? Why can't God reuse pseudogenes to turn it on at a certain environment and then turn it off at a certain environment, right? After the post-flood, several pseudogenes have been turned on and off in order for certain genes, okay, have been turned off and they are called pseudogenes, and some were turned on and they are functional. Okay, the same designs for functional pseudogenes, or let's say in a large scale, or let's say,
Well,
or
what you're saying is that many of these non-coding RNA genes, for example, pseudogenes, it applies to a lot of the non-coding RNA genes, but in this specific case, the pseudogenes, they're only expressed or they're only turned on
when they're needed.
in different environmental conditions. Exactly, when they're needed, when the function is necessary to the specific organism. So a lot of times those pseudogenes are turned off, and this is a quick rebuttal to so you'll find that evolutionists like Herman Mayes, they'll point to certain sections of the mouse genomes that are knocked out. Boom, 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 they're snipped out.
Yes.
And the mouse develops fine and lives its life fine. So now Herman Mayes, for example, will say, see, those portions were absolutely useless junk. And they'll think that that's proof against our prediction that the majority of the genome is junk. But as you're pointing out, and Dr. Herman Mayes is not realizing, many non-coding RNA genes, like the pseudogenes you're talking about, they're only expressed and turned on under certain conditions. And a lot of those conditions, guess what? They are not necessary, and they are not available in the lab type settings. And then we could obviously go into redundancy as well. That's incredibly important. You snip out a gene and another gene backs it up. We have tons of redundant elements and mechanisms in our genetics. But yeah, that's a really good point, that a lot of those pseudogenes, if they're not necessary, they're going to be turned off. And if you snip that out in a gene knockout test, well, yeah, the mouse will be just fine, because that gene, for example, wasn't needed in that moment. It wasn't turned on. So good point, Jim.
Okay. Yes, the mice that they tested, actually, how many years old is the mice right now? Those knockout genes have not. I mean, the mice that they knocked out have not lived their entire lives yet. So we don't know what will happen to them in the future. Will their life expectancy be reduced, or what will happen along the way? What disease will they develop? So that is something that has not yet been tested. Okay? So they're looking at right now the mice. Okay? And now the mice is no longer in the lab. They release the mice. You know that's something for truth? Because they don't want to go further from their initial yeah, from their initial discovery, because they have a feeling that this junk may perform something. And there's an article by who was that? You did share it in a debate. Who was that again?
You mean Dr. Jeffrey Tompkins, his article regarding a
Yes,
paper with, yeah.
yes. Will you please there's already inconsistency. There's already a problem even while the mice was in the lab. So what is it again, Sunny, for truth? I
I
forgot.
believe
So
it, I'd have to pull it up. I believe it was they were snipping out sections of long non-coding RNA genes, and they ended up having detrimental effects on phenotype, genotype. I believe it was effects in the testes, in the immune response. I could be wrong about that one. I'd have to pull it up. But yeah, it was Dr. Tompkins was going over a
yes.
specific paper regarding one of those tests. But I believe it was long non-coding RNA genes that were. It showed those knockout tests showed lethality in developing tissues. So I know that the colon was one of them, the brain, the stomach, even the heart and the lungs. So these were not, and even the ones that weren't lethal, that just killed them right away, they at least caused severe growth and developmental problems. Even in brain formation, the early stages of brain formation, there was seen difficulties. So I'm glad you brought that up. That's a really good point.
So the environmental factor is one. and this Jeffrey Tompkins is another. So what you said about those problems that Dr. Tompkins was able to dig dig the correct English, was able to find out, would predict that the mouse will not reach its average life expectancy. Correct?
Exactly.
Right. That's what I said before. Okay, let's move away from the mouse.
And I want to point out one other thing, actually, because for the audience, and a lot of this is technical, and as you can tell, we just spent half an hour just on the hierarchies seen in pseudogenes, for example. So everything Jeremy is saying really is, for one, it's a really good representation of the evolution model, and it's a really good refutation of it as well. But I know it's technical, and in a nutshell, for the layman, if we can prove, and obviously there's a lot more details than this, but just for the listening audience who might think it's too technical, the bigger picture of this is this: if we can prove a near fully functional genome with certain genetic markers, for example, as highly functional genetic elements, then that overturns those phylogenetic systematics, that overturns those nested hierarchies and turns them into hierarchies by design, as we've always been predicting. But another argument by Herman Mays as well, we just refuted one of his arguments, but another one he'll say is the onion problem. You know, why do onions have a lot more genes or bigger genomes than humans? Or why do certain lizards or reptiles or amoebas, for example, or plants? They'll point to, oftentimes it's plants that they'll point to. Well, and they'll say, you know, obviously most of this must be junk, because why would a plant have more in its genome than a human? Well, here's the
answer.
I'll refute that real quick, and then Jeremy, you give me your thoughts too, because you're really good at this. Well, for one, plant genomes, they have to be bigger for adaptive purposes. Okay, yes. Adaptation is extremely important in plants. So just think about it, and Jeremy, you think about it as well. Like plants, for one, they can't move. So let's say animals, insects, rodents, fungus, temperature fluctuations, even pH changes. Based on, say, acid rain, for example, you can name it. You can name it all. It's trying to kill the plants. They have to constantly adapt to a variety of conditions, as I just said, especially in the environment. So if they cannot adapt instantly, what happens? These plants die. Boom, they're gone. They can't put on a jacket like us humans. They can't adapt in these types of ways that we do. They can't even move underground like an animal does. They can't burrow away from a negative environmental condition. So guess what? They adapt or they die, which means they need a bigger genome for what? Backup redundancy. Redundancy is good in computer code, and it's phenomenal in our genetics, the DNA code. So the answer in short is redundancy. What are your thoughts on that, brother Jeremy?
It's simple. They are exposed. That is why they need more genes. They are exposed to the environment, like you said. They are exposed. They don't move. So exactly. Yeah, they are exposed. We are not exposed. When it rains, we can go. We can hide.
Exactly. If it's cold, we put a jacket on. An animal, a lot of times they burrow. Birds migrate, for example. Like there's so many different defense mechanisms, but plants, they just sit there. They can't move. They have to adapt or die. That's the key. And redundancy, I ask it in every single debate, and even your big shot evolutionists. Like I asked RJ Downard in our last debate. It's like redundancy. DNA repair mechanisms, Jeremy, as you know, it requires forward thinking. And Jeremy, do you know that natural selection acts in the short term, but redundant mechanisms and elements, guess what? Those are beneficial in the long term. It's kind of like, how does natural selection evolve a spare tire? You know, if an evolutionist were to look at a car and assess it and they find that spare tire in the back, they're going to be like, well, what is this? Well, it's already got its 4 tires. You know, this is just junk. This is just unnecessary evolutionary leftovers. No, that spare tire is there for purpose. If you get a flat tire, right? If the car experiences some negative weather, and boom, flat tire. You go over a nail or something. There's your flat tire. Your backup tire, and that's what we see in our genetics with all this redundancy. It's phenomenal. They can't explain how evolution would evolve that, which it requires forward thinking. We can, and plants, boom, the onion problem. It's not a problem, and it just makes sense that plants require more adaptation, adapt or die when it comes to plants.
Yeah, I just sorry, you were saying. I'll let you finish.
Oh no, that's it, brother. That's it. Go ahead.
Okay. There's also a gap in the genome between plants, onions, as compared to other living things like birds, the land creatures, and us humans. There's a gap in the number of genes overall. Now, why is there a gap? Okay, that's it. You said because they are exposed, and those birds, those land creatures, those sea creatures, they can hide from certain environmental conditions. So though they do not need a larger genome. Okay, so there's a gap. If you look at the number of DNA letters between those plants, onions, compared to all land creatures, sea creatures, and humans, you will see a gap in the number of nucleotides, in the number of DNA letters overall. Okay, I'm going into specific now, the numbers again. Okay, the gap is huge. Okay, the onion human gap is huge. Okay, imagine the other plants in which are more exposed, more vulnerable than onions. Okay, so the gap gets bigger, and it gets worse for evolutionists. So if so, so that's it. Okay, there's a genetic gap. I mean, genetic gap simply the number of nucleotides, DNA letters. Okay, are we on the same page here? Standing for truth.
We are, and I just wanted to point out that a lot of times, if for a creationist who's just even a creationist who's not extremely technical. That's why I went over the bigger picture of it all. You know what I mean? We can go into all the very small, minute details about what's functional and how it's functional. But realistically, it's as simple as if we can prove a near fully functional genome, it turns those hierarchies, which appear to be reflection of descent with modification, into that which is hierarchies by design. And that's why it's important to go over their so-called best arguments, because they need to hang on to junk DNA, as you know, Jeremy, because without junk DNA, evolution by common descent is absolutely destroyed. They need a genome that is max 20-25% functional, because they need those junk areas in order for those mutations that accumulate to be absorbed by those areas. Right? Those areas need to take these mutations that are accumulating, keep them as a reservoir for genetic and evolutionary change and advancement in the future. That's why they say they're neutral. But the more functional our genome is, not only does it prove our model of created heterozygosity, it also proves and demonstrates that our genomes are degenerating much faster than anybody ever thought, because now those neutral mutations that they say, they're actually nearly neutral. They only have very subtle effects on fitness. And those over time, they will accumulate, because nearly neutral and neutral, same thing. They're invisible to natural selection. Now, the evolutionists want those to be absolutely neutral. No effect on fitness at all. That's what they want. That's what they need. But the truth is, the more functional the genome, those neutral mutations are nearly neutral, but they're still invisible to selection. So therefore, they're building up, like, for the audience sake, and a good analogy. My favorite analogy: it's like a single spelling mistake in a book the size of an encyclopedia. One single spelling mistake is, at the end of the day, it's inconsequential to the message of the book. But the build up over time, without any correction of all these spelling mistakes, it's going to degrade the message and ultimately degrade the book. And that's what we're seeing in the human genome. So go ahead, brother, go ahead.
Okay, to put it in a nutshell for the audience also, I think standing for truth made it technical, right? You did make it technical. So to put it simply, they need a junk DNA because evolutionary history is long time scale, right? So if a car, for example, we love to use the car, don't we? Standing for truth.
We do. We do. We love
The
our
car, for
cars.
example.
Yeah, but Jeremy, cars don't reproduce. Remember?
No. I'm just giving an example, my friend.
I'm just pretending to be the evolutionist. How many times have you heard that, Jeremy? Cars don't reproduce.
I heard it all the time.
How do you respond to them when they say something so silly, as if we weren't aware of that?
Because we say that that it all comes to genetics, right? So
Right.
they would say, why would we use the car as an example? It's just an example. Come on, evolutionists,
It's an
dumbass.
well, and
Um,
here's what I find
for
funny
sake
too,
of,
Jeremy. Real quick, sorry to cut you off, but what I find funny is every time they say that, I always respond by saying, "Hey, listen, adding reproduction, adding complexity is no help to you. It makes your problem that much worse." And it's also, Jeremy, I think it's important to point this out to them, and I often do. See, we're doing our best to represent their model accurately. When they say things like that, they're fighting against a straw man. Because Jeremy, what do we say? We know from the Bible that the Bible says we are made in God's image. Okay, that's where we start. Now here's our model. Okay, if we're made in God's image, okay, now we need to make retrodictions, predictions. Okay, maybe we can get a sense for how God designed the biological world by the way we design things. Now, guess what? Guess what? By accident, we don't even do it on purpose. These engineers at these big time car factories and companies, they're not saying, hey, let's prove the creation model and just so happen to build in nested hierarchical patterns. No, they do this by accident. So this is just confirmation of our model. Now, if it just turned out that we're just building randomly and there's no type of hierarchy, well, then they may have an argument. But that's the point, is we accidentally build in these types of patterns. Therefore, as we suggested, maybe this is the way that God designed life. And it just so happens that we design in hierarchical patterns and God designs in hierarchical patterns. It's a beautiful fit.
and does it extend between the families?
The hierarchies extend between the families, but as you said, there are gaps. There are ways where we can show that not all life is related, as they would say, based on the hierarchies. But I wasn't getting into that. I was just showing what I believe to be the best response to an evolutionist who says cars don't reproduce. You know, I think there's two ways you can go about that, both being the ways that I just explained.
Okay, all right, I understand. Now going back to the car and the evolutionary timescale, another reason, just to complete what you said, standing for truth, my friend. Why do they need junk DNA other than the reason you suggested? The reason they need junk DNA is because hang on, there's a music that is interfering. Hang on, just 10 seconds this time. Why do they need junk DNA? Because, just to finish what Standing for Truth said, they need junk DNA because evolutionary history is a long time scale, millions of years. Okay? And again, we go back to the car or airplane or anything, even a computer. Okay? As the years go by, they degrade, right? To the point that the computer, right now, let's not use the car, let's use the computer. Okay? So the computer will degrade to the point that it becomes what? Junk. Okay? So millions of years of evolution, okay, would create a lot of junk. All right? Just like the computer, if you use it for a couple of years, let's say 5 to 10 years, it will degrade to the point that it cannot be used anymore. Even if you repair it, it cannot be used anymore. It becomes junk through the years. So evolution through the years, through their millions of years, okay, expect junk because it's millions of years, just like the computer. What is the life expectancy of computers, right? So they predict more junk. That is their prediction. The junk will increase actually. No, did you not know that? That was the prediction. The junk will increase.
Right. It
Yes,
has
the
to.
junk will increase. Okay? Just like the computer. It will degrade more. It's as simple as that.
Well, that's why
Okay?
you pointed out before, Jeremy, that evolution didn't predict this many conserved regions in our genetics, because these genes that are conserved suggest function, right? That's because natural selection is preserving those genes because they're important. So why do we see so many conserved regions between, I'm just throwing this out there, let's say the mouse genetics and the human genetics? You pointed out very brilliantly before that evolution never predicted that. They were caught off guard.
Yes, exactly. Then they keep on changing. First time, in the 70s, they predict uh, only 1% are functional. And then after the Human Genome Project by Dr. Francis Collins, one of my favorites, although he is an evolutionary creationist, after the Human Genome Project, which is he led, the function went up to 2%. Okay, that was not the original prediction. The prediction is the genome should go down below 1% function. Okay, because we only need, they say that we only need a few genes, 22,000. Okay, about 22,000 if I'm not mistaken. Okay, protein coding genes. Okay, so it became 2% in the year 2000, 70s 1%. It stuck for so long, and then in the year 2012, no, in the years that follow, the function slowly increased. Okay, ENCODE phase one. ENCODE is the encyclopedia of DNA elements, which maps the human genome. Okay,
Right.
at first, ENCODE phase one revealed they checked 1%. They dare check. They are neutral. Okay, because they wanna research everything. Because we have already studied the protein coding genes well enough, but not enough. But sorry, we have studied the protein coding genes already. It's ongoing, and we want to study everything. We want to study where the diseases come from. So, where do diseases come from? Logically, okay, think about it. Where do diseases come from? From garbage, right? Right? We get disease
Right.
from garbage, right?
Right.
Okay, we get this. That's just one example, of course. So we do get disease from other sources, of course. Garbage is one. I did get a disease from exposing myself to garbage when I throw the garbage, because in my country the garbage is really messy. I can even smell it right now outside. This is how bad this country is. Forgive me, my fellow countrymen, if you are watching. Now, so garbage, right? Garbage. So that is why ENCODE phase one, with the direction of Dr. Francis Collins, suggested that we test 1% of the junk area of DNA, just 1%, to find out if we can find disease over there. And when they checked the junk DNA, the 1%, they found out that 100% of it is functional after all. They did not find disease there. It was only later after ENCODE phase one that they found disease. Okay, and we expected to find disease in the junk areas, indeed. Okay, that is they usually find it outside of the 80% of ENCODE phase two, those disease. Okay,
Right.
so slowly but surely after ENCODE phase one, they were shocked that the entire junk area the 1% junk area that they tested were fully functional. So they continued their test, and slowly but surely, they went into ENCODE phase 2 and found out specific biochemical activities over on 80% of the genome. And they did that, and they did not find the diseases on. They did found some diseases in that 80%, but there were more diseases found in the rest of the 20%. Do you know this? Most of the disease are in the junk areas, just like the garbage where I got a disease one time. Okay, so you would expect diseases coming from viruses, and those viruses, bad viruses, are like the dirt or the smell that garbage produce. Okay, am I making sense? Everybody following? I hope. I'm trying to simplify this. Okay. Now, where do you want me to go, Standing for Truth? Do you want me to continue on the on and off of the pseudogenes? Because there is another argument in which I suddenly remember about that.
I do.
In which
I do. I just wanted to point out too, because what you're saying is so fascinating. Because, for one, okay, you'll find that evolutionists seem to think that RNA products that lead to proteins are all that which is functional. But the fact is, and what ENCODE has been revealing, not all RNA products of the genome lead to proteins. There's these RNA products, for example, they're being utilized in a number of fascinating ways for structural purposes, and I mean just function after function. And the fact is, there's actually over twice as many, Jeremy, and you probably know this, twice as many long non-coding RNA genes as there are protein coding genes. So these evolutionists seem to think that protein coding genes, those are the only ones that's functional. No, not even close. Most of these long non-coding RNA genes that don't even lead to proteins are even more. They're even more cell and tissue specific in their expression than protein coding genes. It's kind of like if you go to a theater and it's a live show, for example, you got all your actors and actresses on stage. Those are your protein coding genes. They're doing a number of things. But guess what? Behind closed doors, you have your set managers, you have your individuals that are running the lights, for example. You've got those that even wrote the script for the production, for example. There's a ton of roles behind the scenes, and these non-coding RNA genes, they have numerous unimaginable roles behind the scenes. You know, they're helping the protein coding genes, for example. So this is what ENCODE has discovered, and it's yeah. I mean, do you agree with that? Do you agree with that, Jeremy?
Yeah, I agree with that. There's another argument to that. The introns and the exons. The managers, right?
Right.
Which one are the managers? Introns or exons?
The exons are the protein coding gene. So
The introns and exons. Yes. They're the managers that you're talking about. Yeah, we have not even. Are we going there? Probably not. But what you said can be applied into those introns and exons. Okay, and there are more things. Forgot. There are more.
Those introns.
Those activation. What do you call that? There are activators over there too.
Right.
Yes.
And as you, well, I mean, at the end of the day, these non-coding, these non-protein coding RNAs, they're heavily involved in regulation, and they literally regulate in nearly, pretty well every single aspect of gene expression. And the introns you're talking about, they are involved in, well, they're rich in these splicing factor recognition sites. And alternative splicing, boom, you got it, Jeremy. And you're brilliant on that topic. So alternative splicing, literally, the best way I can, so you got a single protein coding region of DNA, okay? And as a result of the alternative splicing, this single protein can give rise to hundreds, even thousands of different proteins. How
Yes.
did natural selection and evolution evolve? I mean, we were talking about how did natural selection evolve redundancy? How did natural selection and evolution-based mechanisms evolve something like alternative splicing? It's science fiction what's going on in the genetics.
Yes, exactly. Okay. Are we done with that? Anything
Yeah.
you want more to add? Okay, so
Yeah, well, I just want to point out to the audience, we just really wanted to emphasize the function because that is a direct prediction of our model, and that is what ultimately overturns these hierarchies. Because it shows that these patterns are not truly reflective of descent with modification. They are
yes.
reflective of function. Now, evolutionists, I just want to, and then you take your time, Jeremy. Remember, evolutionists assume all DNA differences, okay, nuclear DNA differences, mitochondrial DNA differences. They assume that every single one of these DNA differences is the result of mutation. So when they see differences in, say, these highly conserved mitochondrial DNA proteins like the cytochrome C, cytochrome B, the CO1 gene, they believe that those differences, okay, are a reflection of time. So you can look at the cytochrome C gene, for example. They're going to say that because the cytochrome C protein is nearly identical, and these are oftentimes referred to as
Just like the gulo.
housekeeping genes. That's what they call them, because they're typically genes that are required for the maintenance of just basal cellular functions. And that's their specific role, and that's their role for all of life, all of biological life. One role, they'll say. But the point is, they'll say that, okay, because the cytochrome C protein is nearly identical in humans and chimps, that's because they are closely related. They share the most recent common ancestor. Then they'll show the differences in that highly conserved region between, say, humans and the old world monkeys, or humans and fish, humans and dogs. And those differences grow larger and larger. And they'll point out, they'll say, okay, why did God make so many different variations of this cytochrome C, which is just involved in the electron transport chain of mitochondria? Okay, it's part of that pathway for the synthesis of ATP. So why do we even see those groups within groups, nested hierarchical patterns, in something like a highly conserved mitochondrial DNA protein, which just has one basal function? Because from a design perspective, you would think that they should be identical. But why are there so many differences across all forms of life? And why do they reflect what seem to reflect nested hierarchy due to descent, right? Because they're saying, okay, these variations in that one mitochondrial DNA protein, that is because different divergence times, different speciation times, for example. But natural selection preserves these proteins because they're important, right? Obviously, if you get a mutation in a cytochrome C protein, that's incredibly important for like energy production, which ultimately is important for the heart. Well, naturally, you're going to die. So ultimately, these are preserved. That's why they want to look at the most conserved genes to build their phylogenetic tree. And obviously, we have an answer as to how this differentiates between the model. But yeah, that's what they point to. But is that, I'll just give the answer here before you go. Is that really, and it comes down to function. That's why we're saying function is key, because is that really the only function the ATP synthase performs, for example? And you've heard Jeremy and I talk about it over and over again, something called protein moonlighting. And Jeremy and I like to put it into an analogy. Think about the human body. Think about all our bodies, praise, your body, the audience, as biological construction projects. What do? We start out as a single cell, and then our entire bodies, every single organ, every single cell Everything that makes us us is built in the womb so that the human form is apparent at birth. This is fascinatingly complex. Now we know this is just basic science. Proteins, what are they? They're one of the main tools for building the body, okay? They're heavily involved in this process from single cell to birth, of course. But protein moonlighting, on the other hand, Jeremy and I often compare it to a multifunctional tool. What human engineer, and I like this because Dr. Jensen points this out, he'll ask, what human engineer builds a device that is not only a hammer, but it's also a saw, a drill, and 15 other things on one device? I mean, that's science fiction right there. What's going on the, real quick, that's going on the cellular level. So this fact of protein moonlighting just shows the complexity in our genetics. Now, here's what's important. Nuclear DNA proteins, that's primarily where we're seeing this protein moonlighting, okay? So now we're making testable predictions on these mitochondrial DNA proteins, okay? That those differences also reflect function. Multiple functions, for example. So not only are they participating in electron transport and energy production, maybe they're also functioning in cell optimization or metabolic optimization. And this is a very, very legit prediction because of what we know regarding protein moonlighting in these nuclear DNA proteins. So, I mean, we're just in the infancy of genetics. And guess what? This is what's important, okay? What model is making the most accurate and what model is making the best testable predictions? That's who wins this war. And right now, it is the creationists, because I've been in enough debates where I've asked the evolutionists, what predictions can you make? And I never get an answer. That is key. Jeremy, take it away, brother.
Okay, I was lost. Where did I end my? Yeah, I did complete some of the holes in what you said already. I believe, like the long time scale. That is why they need the junk DNA, right? So I hope we were able to simplify that, right? Am I able to simplify that, standing for truth?
Of course, brother, of course.
Okay. Yeah. Now I remember the turning on and off of certain genes. Okay. Okay. Of course, it is not good to say that God would create non-functional genes. Okay, it is not good to say that. But of course, after the sin, things started to degrade. Okay. Uh, um, these shared pseudogenes, these shared mistakes. Again, these pseudogenes. What am I about to say? I was lost. You talked too much, standing for truth. I forgot. Forgive me. Uh, Shared mistakes, those pseudogenes, can come from independent turning off or in activation situations or events that occurred after the creation. Okay, like I said before, God, why not? Would He not reuse certain genes? Okay, so as you can see, this relates to the on and off. When you reuse, then you turn it on. Then when you donate it, you turn it off. On, reuse. Off, then you recycle it into something else. Okay. So why would God then not create genes that can turn on and off? Okay, so this is the argument that we can use against the turning on and off of genes from Dr. Dennis Venema. Okay, now, what is that picture standing for, truth?
I think it's praise. Praise, are you sharing screen, brother? This wouldn't be the first time he's shared what he seems to be looking at. Praise, unshare your screen, brother. Yeah, continue. Actually, if there is anybody in the audience, because we are getting to the point where if anybody else wants to join, because we've been doing this for
Yeah, if anybody wants to ask
hours,
questions.
and that's why, Jeremy, because we've been at this for a while, that's why I went really hardcore into the Cytochrome C, because Cytochrome C is important, which I just feel confident that I destroyed there, because I oftentimes see creationists, they'll just say, like Stefan Frello points this out a lot. Herman Mays points this out a lot. Conspiracy Cats points this out a lot. Why did God make so many different variations of the same important gene? Why did God make it look like evolution by common descent? And oftentimes creationists will just say, well, you know, common design. It's not really answering the question, because they are pointing out a legit concern that really does seem to look like common descent. You know, that would be a differentiating evidence, which means we have to have an answer to these types of questions. And you hear it from these top evolutionists like Stefan Frello, Herman Mays, Conspiracy Cats. And I've heard creationists respond with, okay, maybe God did create them all the same, but since it's been 6,000 years, they've accumulated mutations, and that explains the differences. Well, I wouldn't recommend that argument, because that would imply that the mutations that have accumulated since creation still happen to fall in these nested hierarchical patterns. Like, why did humans, chimps, the old world monkeys, and lemurs, for example, why did they all accumulate just enough of the same mutations in, say, the cytochrome C protein to look like evolution by common descent, right? So I don't believe that that's a very good answer. The best answer is the fact that the DNA differences, they assume, are the result of mutations. We don't. We assume the vast majority of DNA differences are the result of created functional diversity. Therefore, we're going to predict the differences are functional. And that's why function is key. That's why I emphasize function, function, function. From a design perspective, why would the, say, zebra ATP synthase proteins have a different mitochondrial DNA sequence in nucleotides than a toucan, you know, like a bird, for example? Well, of course, if these proteins, these highly conserved proteins, are multifunctional, well, cell optimization, metabolic optimization will be different in a zebra than it will be in a bird, or it'll be different in a human than it will be in a lemur. That's why those differences exist, for functional purposes. So one, to the listening audience and anybody interested in apologetics, don't just say common design. It's not a good answer. And two, it wouldn't make sense that God created these highly functional conserved mitochondrial DNA proteins the same, because they do reflect these hierarchical patterns. Now, as I pointed out in the beginning, we both expect hierarchical patterns. Boom. That's agnostic. That's neutral to the debate. What wins the debate? What wins the war? It's function. One word, function. And that's why Jeremy and I are getting so technical as to the types of functions. And that's why we used ENCODE, for example, or as Jeremy talked about, the slight nucleotide differences and even the pseudogenes. You know, so this is quite a deep topic, but it's fascinating that we're the ones making the predictions. Jeremy, I want you to talk next, but I do want to make sure we're hitting everything. That's why I'm kind of jumping in and just talking really fast, because I guess that's one thing I can do pretty good. But we have to cover Herman May's third position codon variation, but we got to do it quick. So unless you want to kind of just go into it quick, give an answer quick, kind of like I'm doing with the Cytochrome C, just so we can, Praise wanted to bring more people in for more of an open discussion. So I'm going to hand it over to you.
Take over the Herman Mays thing, and I'll provide specific numbers. Like I said in the start, I will provide specific numbers. I will not use function specifically. Okay, function is yours. I'll use specific numbers.
Okay. So do you want me to go over the Herman Mays argument? This will be the only one that we need to cover. We've demolished everything else. I'll give my explanation in terms of function, and then you'll give yours in terms of nucleotide differences. Is that how you want to do it?
That's what we discussed at the beginning, right? So yeah, okay. I just wanna, because one of the common arguments of evolution is like By the way, to put your argument recently in a nutshell, um, yes, the families can share. In a way, in another way to look at it, the families can share the same higher. The families can share the same hierarchical patterns as well, not only at the animal kind level, but at the family level. But to simplify what Standing for Truth said, um, those hierarchical patterns not only should be functional. You're forgetting the Standing for Truth. They not only have to be functional, but their function has to be different. For example, the cow. Did say that. Okay, I'm sorry. So I'm just trying to make it easier for the audience to understand because you were going
Well,
so
that's
quick.
why. Well, that's why I was explaining that the cell optimization and the metabolic optimization in these highly conserved mitochondrial DNA proteins as secondary functions versus just your basal function that these ATP synthase proteins have. Those functions would be different in, say, a human versus a chimp versus an old world monkey versus a fish versus a reptile, because a reptile is going to have metabolic and cell optimization that is different than the chimp or the human. So
So in
these
short,
functions have to be different. That's why I point out that these genetic markers that they use, those are what really need to be highly, highly functional.
the
Specific
audience,
to your
just look at.
kind.
Sorry, it's my turn. Okay, audience, look at my hands. Each of my hand, let's say what here is a specific animal kind. Okay,
Right.
these specific animal kinds. Uh, if you wanna, if there is a hierarchy between the families. Okay, one, these hierarchies between the families, let's say the cow and the let's say whale. Oh no, the horse and the whale, because they say that the horse evolved into the whale, or the whale evolved into the horse. I forgot which one evolved first. Forgot. Sorry. Um, if they share, because they are it's, one is land dwelling and the other is sea dwelling. If they share a nested hierarchical pattern, okay, in order to disprove that, you would need them to not only be functional, but their function has to be different. Okay, that goes the same with the other animals. Okay, if they share the same nested hierarchical pattern, and like I said, those nested hierarchical pattern, I said it almost at the beginning of this video that there are gaps within those hierarchies. Now, okay, my turn. Standing for truth. The argument, we're gonna make this comprehensive and complete, correct? So the evolution, because I was not able to finish the reuse on and off thing, okay? Because it's not thoroughly refuted, okay? All right, they have their worldview and we have ours. So how do you disprove the on and off that evolutionists are saying? Now, one way to disprove the on and off of certain genes, they say in evolutionary history, it was off at this 100,000 years ago, then it turned on 1,000 years ago. That is why we find function. So this is one of the counter arguments to your function argument and Dr. Nathaniel Jeans's argument standing for truth. Now, in order to get around this argument from Biologos, well, I really hate um, going against fellow Christians from Biologos, but I have to because, well, I have to.
And as you know, Jeremy, scientists such as Collins, Vanema, they are some of the smarter evolutionists out there. And you oftentimes find, what I find funny here is we're giving 99.9% of the evolutionists their own best argument, because I've had 70 debates and only like maybe a few of the evolutionists actually know how to utilize their own best evidence. But people at BioLogos like Vanima, Collins, they are aware of these hierarchies and they
Yes.
utilize them, which means it's important to, it's as Todd Wood says, we should never underestimate the enemy, the evolutionists, for example, because your 0.1% of the evolutionists that are, I guess, trained enough and well-read enough to use these types of arguments, we have to have good refutations and good rebuttals. It's important.
Now here I'm gonna teach Young Earth Creationists how to answer the reuse argument, the on and off of certain genes I explained a while ago. Okay, standing for truth, you know about this. Now, okay, first of all, why not would God reuse certain genes? Huh? Why not? And turn them off when they're no longer needed? Huh? Okay, so that's one argument you can use. Why not God? God can also reuse by turning it on and turning it off. So that is one argument you can use. Okay, it makes sense, correct? Okay. Now, many of the claimed pseudogenes are not really pseudogenes. This is the second thing that you could tell evolutionists, atheist evolutionists especially. Okay. Now, to counter this argument, almost every atheist, almost every evolutionist, whether it's a theistic or atheistic evolutionist, accept this reuse argument that I mentioned earlier, which the young Earth creationists can also use. Like I said, that God reuses junk genes by turning them on when it's needed. Okay, because we found many junk genes to have functions after all, so they turn off. So ENCODE kind of like revealed, and many independent papers revealed that some genes that we thought were turned off were actually on. Okay, so this is the second argument you can use. All right, and certain pseudogenes, like I said before, lack function. Okay, they are degenerate. This is the third argument you can use. They are degenerate, but it doesn't mean they are not functional. Okay, a building that is old, which looks degenerate, okay, you can still live there. Correct? A car that is rusty and old, you can still use it, right? So a pseudogene that is degenerate does not mean it is not functional. We have to be clear on that. Do not be misled. Okay? Lack of function does not mean no function. Okay, that is the third argument against the owning and off of genes. Sorry for my English. All right? They are misleading people, especially atheist evolutionists. But Dr. Dennis Venema of BioLogos, Dr. Dennis Venema is a Christian, is honest enough to admit that the lack of function, okay, does not mean it is functionless. It's in his book. Anybody read this book? You can confirm me. You can confirm it right now. Okay? Okay? Plus, if pseudogenes are really damaged genes, they can occur independently. Not the nested hierarchies evolution is suggested. Why are these pseudogenes everywhere? Okay? Why are they everywhere? They do not form the vast majority of them do not form the hierarchy that evolutionists are expecting. Okay, I can name hundreds of them. Okay, it is the evolutionary preposition, the evolutionary bias, which makes them see nested hierarchies. I'm sorry.
Oh, I'm just saying that's a good point.
Yeah. Sorry.
Yeah, I was just saying. No, I was just saying that's a good point. That's a very good point.
Yes. Okay. An evolutionary preposition or bias, if I'm not mistaken with my English, makes them see nested hierarchies in pseudogenes. And I already gave 4 evidence against pseudogenes, which young Earth creationists can use. Okay? Okay? Hear me, young Earth creationists out there? Okay? You've
Well,
never
not
heard
to
this
be,
argument.
and Jeremy, I wanted to continue. I just want to make one little point to the pseudo. I'm not sure if you made this. We kind of talked about this with the conserved genetic regions, but the fact that these pseudo genes are so persistent and they're found everywhere, like you said, that alone is at least additional evidence for their activity, because just like the conserved regions, just like the 80% activity that was revealed by ENCODE, that is a very serious problem for evolution. They never predicted that amount of conserved functional genetic regions, because natural selection, okay, it should remove this type of DNA if it were actually useless, or just evolutionary leftovers, or genetic mistakes, for example, because the DNA that's actually manufactured, okay, by the cell is energetically costly. It's not just going to replicate and manufacture this useless, non-functional DNA for millions and millions and millions of years. So because of the clear lack of selection, the selective pressure, I should say, on this type of DNA, that should reveal to anybody that there's some activity going on. Because, yeah, we don't have absolute demonstration and proof of every single function in these pseudogenes, but the persistence of them, in my opinion, is evidence for their useful activity, because natural selection would have removed all this junk long ago.
One more thing standing for truth in the audience. When you say pseudogene, pseudogene consists of several genes sometimes, okay?
Right.
Right, okay? And do you know, just like you debated before about the chromosome 2, in which the middle has a certain function. The
Functional. Yeah,
functional
like the site is a promoter for
element.
the gene.
Yeah,
Right.
it's the same with pseudogenes.
Right.
One
Right.
pseudogene or two or one pseudogene, a collection of pseudogene, it can be interpreted as one pseudogene. One pseudogene, okay, for example, there are parts of that pseudogene that has function also. So how can you
Right.
call that a pseudogene if there are functions over there? You can call that degenerate, but you cannot call that pseudogene anymore. There are many pseudogenes in
Are
which
are
part of
those
it,
are those the parts of the pseudogenes that they would perceive as like the disablement parts of it? The parts that.
disabling parts. No, I'm talking about the functional parts of the pseudogene, just like the functional parts of the telomeres,
Right.
the
Right.
centromeres of the chromosome 2 fusion.
Okay. Okay. I get
That's
it.
the
I get
one.
it.
The chromosome 2 fusion is like the pseudogene. I'm just trying to compare. It's like the pseudogene. There is
Right.
a function there, here, there, here, there. And a pseudogene, there is a function here, there, here, there, here, there. So
Right.
you cannot really label them as pseudogene. It's
Right.
misleading. The fifth argument young Earth creationists can use against the owning and offing of certain genes. And the first one was, like I said, why can't God reuse certain genes? And when he reuses certain genes, then that it means it is being turned off, and it is being turned, and when it is not needed, then he turns it off. Okay, so we have 5 evidence against the evolutionary argument for their pseudogene. Don't forget that. Although it's true that some pseudogenes are all really pseudo, okay, no function, okay, but many pseudogenes also have function within them, and you cannot call that pseudogene. It is the evolutionary proposition that
Right. It's their presupposition that they're broken.
yeah. Now let me. Can I go back to the olfactory receptor? Because
Yes,
that was
I just want to point out the fact that I find that it's unfortunate. It's kind of like the term microevolution, when we know it's literally just nothing more than basic adaptation. They've invented the word and we're kind of stuck with it. just like the endogenous retroviruses or the retrotransposons or the pseudogenes. They invented it. We're kind of stuck with it because oftentimes you'll find me referring to them as DNA elements, for example, because if you have these so-called quote unquote pseudogenes that they're saying are just broken genes and now they're shared among the primates with proves common descent apparently. But if those genes, and we know this, we've been talking about it, if they are necessary and required to sustain healthy life processes in the cell, can we and do we really have to keep calling them pseudogenes? You know, because a lot of these that the evolutionists have referred to as fossil sequences, that's pretty much what they are, genomic fossils, right? They look at the fossil record for our so-called ancestors. Now they're looking in our genetics for our so-called fossil DNA sequences from our fossil ancestors, which is funny. That's their presupposition. But now we know these are highly functional. And I think it's better to call them DNA elements, like just like your retrotransposons, your ERVs, your ALUs. I refer to those as a variation-inducing genetic elements because once again, those, well, they're known to induce variation in genetics, and they are also highly functional as DNA regulators and things like that. So I just wanted to point out that one. I just wanted to point that out for the audience there, Jeremy. But yeah, go ahead, brother.
Okay, now going back to one of their best evidence, the one of the on this nested hierarchy thing, okay, not overall, because their best evidence overall is distant starlight, in which we have debunked yesterday,
right,
in 2 hours.
Actually, Jeremy, before you go, I just noticed Sal. God bless, brother. We got Salvador here. He just joined us. Actually, I think you've been here for about 5 minutes, so he's been an incredible blessing to our channel. He's given some amazing lectures. He's a warrior for Christianity. So, Sal, thanks for joining us. I want to point out to the audience, I did say it in the chat, but I want to just point it out here live that Sal, he's giving us his time. Sunday, 9:30 EST, he's going to be debating a gentleman named Ron, both highly educated and intelligent individuals. They're going to be debating genetic entropy. So I can speak for myself and probably the audience. We're really, really excited for that debate there, Sal.
I'll explain the I already explained parts of the olfactory receptor during the early parts of this video. I'll try to make it quick. I hope the audience can keep up so that Mr. Salvador Cordova can also speak. Now, we pay attention to the primate olfactory receptor, like I said in the early parts of the video I referred to a while ago, which are also pseudogenes. Now, do you get? Okay, I will not ask questions anymore. Okay, so this will be quick. What do you call that? Damn it! Sorry for saying the word. Forgive me. The urate, URATE, oxidase. I'm not sure if I'm pronouncing the English properly. You might misinterpret what I really am saying because I am not from the West. I do not speak very well in English. The urate oxidase, okay? Urate oxidase. I hope you guys heard of it. The urate oxidase. OXIDASE. Okay, it's called UO. The urate oxidase pseudogenes, which is an enzyme which is at the degradation area of the primate olfactory receptor. Okay, there is a function in the middle of the degradation area of this oxidase. So is it still a junk? No, it's just degraded. Okay, this is a sample of the olfactory receptor in which they claim is junk, but it is only degraded, so it is not junk. Misleading. They should say it is degraded. When you say a building is degraded, it can still be lived upon. We can still live in it. A car degraded, we can still live in it. It can still be functional. So you say degraded, not functional. Do not mislead people, because this is at the middle of the degradation area. This OD, this this urate oxidase. Okay, it is a they say it is a pseudogene, but no, it is not a pseudogene. Okay, and we even know the function of it. Okay, the function is to prevent the creation of uric acid. Okay, its function is to prevent the creation of uric acid by making it fast. Sorry, what is it English? What is the English of it? By speeding up the oxidation of uric acid by little baby. Sorry, said baby. By sorry for my English. Salvador, do you know how to speak Chinese? You look like a Chinese. I can speak better Chinese.
No, I only speak English. I used to know Tagalog, but I can only
Yeah.
speak English. Sorry.
So salamat na nandito ka, pare. Right?
You
In
said
Tagalog.
thank you, and I'm here now.
Yes, that is in
Okay.
Tagalog. Yeah, in the
Do you
Philippines.
speak Tagalog?
Yeah, I speak 5 languages actually. Now,
Oh, that's excellent.
huh?
I wish I could speak even one. I can barely speak English sometimes.
Where the thing in the English part? I can speak Mandarin, Hokkien Chinese, Tagalog, Ilonggo. You know Ilonggo, right? Iloilo. Sorry. Never mind. Let's go back to topic so that you can speak.
So what is your native language?
What do you mean native language? My primary language is my first language is Hokkien Chinese. That's my first language. My second language is Tagalog. My third language is English. My English is not even my second language. Okay, and then my fourth language is Mandarin, and my fifth language is Ilonggo, or a language from Iloilo in the central Philippines, because I studied there for 1 year. Now, going back so that you can speak. I'm sorry for.
But by the way, thank you for your presentation. I learned a lot.
Yeah, I'm sure I'm
Thank you
gonna
for your
learn a lot from you
presentations.
later. I'm sure I'm gonna learn a lot from you. Sigurado ako na na marami din ako matututunan sa iyo mamaya in Tagalog.
I'll translate. He said, "There's a lot I can learn from you a little later."
Yeah, from Salvador. I can also learn from him. I think I've heard him debate before. Okay. All right. Now, the so-called pseudogene in the olfactory receptor. Okay. The function of this is to prevent the creation of uric acid by speeding up the oxidation of uric acid by tiny oxygen. Okay. Leading to the production of urate. Again, urate. Hard time speaking English here. Urate oxidase and hydrogen peroxide. I'm sure you guys heard of hydrogen peroxide. Now, the yeah, we see like I said before. I'm not gonna repeat it. The function of the chromosome 2 fusion site is a good example. Is a good comparison to what I'm talking about here. Okay, but yeah, okay. But by the way, to the audience who do not know what hydrogen peroxide, sorry, hydrogen what hydrogen peroxide means. It is used for our skin to prevent infection of minor cuts. Okay, when you cut yourself using a knife, burns and more. Okay, cuts, burns and more. Injuries, in short. Okay, forgot what the others are, but more or less they are for injuries. Hydrogen peroxide. Okay. In short, what I'm saying here is so far we have realized even after the great human genome project. Okay, so see, there's function on the olfactory receptor part of it. Like I said, it is the oxidase pseudogene, and going towards the hydrogen peroxide. Sorry, peroxide. 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 Okay. Now, in short, what I'm saying is, so far we have realized, even after the Great Human Genome Project, that we know little about what the vast majority of what the human genes do. Okay, especially the pseudogenes. So pseudogenes have not been fairly analyzed. They have preposition. Okay, so I say they are not fairly analyzed because preposition is added or biased by the evolutionary community. They just assume evolution. There is a preposition. Okay, they have to be fair. Okay, even biologus sadly is unfair regarding this issue. Okay, so since that to be true, Then how can evolutionists create nested hierarchies based on pseudogenes? See, how can they create nested hierarchies based on pseudogenes with these functions and different functions of pseudogenes around, right? Standing for truth. The pseudogenes are spread across different animal kinds. So the function here is different for this pseudogene, and the function here, those pseudogenes look the same, but their functions are different, and they're functional. That is the point. Remember, I told you that a long time ago. See? Okay, so they cannot create a hierarchy that extends between one family to another, right? That is just a little evidence. I can give specific numbers here. Now, okay, let me finish. Let me make it quick, okay, so that Mr. Salvador can speak. Now, the known pseudogene, okay, the function of pseudogenes that we have found out, okay, so far, and there are more. Okay, I'll just name a few, so it'll be short. Now, the known widespread is widespread the correct English. Never mind. I'll not say widespread. Now, the known pseudogene functions that we have found out is transcription. That's one. Another function is it enhances pseudogenes are functional because they enhance. It is the pseudogene functional enhancement. It sorry, my English. The pseudogene functional enhance another function of pseudogene is the pseudogene functional enhancement of immunoglobulin diversity. I guess you guys heard about immunoglobulin diversity. Immunoglobulin. These terms really confused me. Immunoglobulin diversity is okay. It is the enhancement of the immunoglobulin diversity. So it is the second function, and the regulatory role, standing for truth. It also helps regulate. It also regulates. Okay. Some pseudogenes do perform I'm stammering. Sorry. Regulatory roles. Okay. Another function. Pseudogenes can also serve as non-coding functions in islands, as I call it, as they call it. Sorry, as scientists call it, as I recall, which are lines or stretches of DNA that are enriched in. Did you say cytosine? Something for truth? Was it cytosine that you? Yeah, cytosine. The function can serve as a non-coding function in islands or stretches, as I recall. Sorry, islands, as I know, scientists label, which are lines or stretches of DNA that are enriched in cytosine and guanine. Cytosine, guanine, CG is part of the GCTGTA of the 4 DNA letters, the primary 4 DNA letters. Okay, at the expense, and these cytosine and guanine again, these pseudogenes can serve as non-coding functions in islands, okay, which are lines and stretches of DNA, like I said, that are enriched in cytosine and guanine at the expense of adenine and thymine. Also, okay, A and T, okay, part of the 4 DNA letters. So we have C, G, A, and T. So at the expense of adenine, whatever the grammar is, and thymine, thymine. Okay, thymine. Okay, in short, like I said, the GTA. I'm repeating myself. The GTCA of the DNA letters. Okay, enhancers they are, and a type of gene regulatory sequence I mentioned a while ago, and its surrounding regions. It is a DNA structural feature or design, not a nested hierarchy. Okay. So here we go. I disprove another nested hierarchy evidence from the overall families because they contain these GTCA. Okay, now I have to be fair to evolutionists also. So I'll give an example of one of their more reliable evidence. I will not lie; they have some evidence. Okay, good evidence. Now, I think one of their best evidence of this hierarchy, because this video is about hierarchy among pseudogenes overall, is the stop codon. You guys heard of the stop codon? Uh, that is what's that again? I guess I'm sure you guys heard of the stop codon. Okay, uh, it is premature actually. Sorry, I forgot the exact data about that. I don't have a script here. So I just you know. Okay, there are two premature stop codons not known for the urate oxidase, which I mentioned a while ago, pseudogene. There are two stop codons not known for the urate oxidase pseudogene. It is at the codon number. Here we go. Here is how I am going to use numbers against evolutionists. Okay. Good luck, Herman Mays and the others of disproving this.
Are you, Jeremy? Are you talking
I
about the
will debate you regarding this.
Jeremy? Are you
If
talking
you
about the wobble position 3?
No. It is at the codon 43 and 187, as I recall. Do you know about this standing for truth? Oh, never mind.
Well, because
Do
you
you
said
want
stop codon.
stop codon? Yes.
Okay.
It
Okay.
is at the codon 33 and 187, as I recall. Oh, yeah. I finally remember the exact number. It is at the codon 33 and 187. And they do follow an evolutionary shared mistake idea. Okay, members of the orangutan chimp human share this codon 33 premature codon. Okay, this is this is one hierarchy that you seldom hear. Okay, now while one at codon 187 is found in gorilla, chimp, and humans. Okay, okay, there's a one codon at the while one at codon 187 is found in gorilla, chimp, and human. It looks like there is a hierarchy of shared mistakes, correct? Standing for truth, just like your cytosine and the Zulo gene. Now,
Are you saying when you say are you referring to Cytochrome C? You said cytosine.
I I forget. I'm really confused of the term. Okay, so you can correct me if I'm wrong. Standing for truth.
Oh no,
Okay.
it's okay. I think you're right because you were saying it sounded like you were referring back to something I discussed earlier. But what I discussed earlier was the Cytochrome C. But no, keep going. You keep going,
Yeah,
brother.
yeah. All right. I represented the evolutionary idea in specific numbers here. Now I'm gonna refute it. Okay. I'm fair to the evolutionists, and I'm also gonna refute it. Now, in order to refute it, or in contrast to correct English. There are three others at the codons 18 and 1. Sorry. Uh, okay. The first one is 187, codon 187, correct? In order to refute that shared mistake, there are others at different codon positions. Codon 18 this time, and 167, and 19. I wish I can find my archives over there because the specific numbers are there. So like I said, in contrast or to refute it, there are others at codons 18 and 187 and 197, which are unique to one type of primate only. So as you can see, there are shared mistakes. There are also unique elements over there, unique codons. So what is it? Is it really common design or common ancestry? Okay. So more and more tests has to be done. Okay. Okay. And based on evolutionary bias, they imagined. I'm sorry. They okay. Based on evolutionary bias, okay, they are imagining to have come from they are imagined to have come after each primate that have branched off its lineage. That is why the numbers have changed. Okay, but they don't even know the initial number. Okay. Lastly, one of the premature stop codons in the urate oxidase pseudogene, we're using the primates because it is one of the most commonly used olfactory receptor part, has an
Hey,
evolutionary.
Jeremy, I'm sorry to cut you off. Just because we've been going at it for a while, because I'm going to want to, just to make this complete, since a lot of people are requested this specific video on hierarchies. I still have to address Herman May's neutral variation. I can do it in probably under 2 minutes. If you could just finish up what you're explaining real quick, just so we
Yeah,
can.
I'm really doing my best to finish the explanation. Okay, okay, all right. There's a huge evolutionary problem even with their explanation that that the primate has branched off its lineage. That's why the numbers have changed. Okay, it became 18 and 167. Okay, mutation at the codon 107 is more complex than others. Okay, it occurs in the gorilla, okay, the orangutan, and the gibbon, gibbon, whatever, and therefore it needs it requires multiple origins of this nonsense mutations. Yeah, I call it nonsense mutation. So again, a hierarchy, as you can see. Okay, now worse for evolutionists. Okay, so sorry, so it is against a hierarchy, as you can see. Okay, now worse for atheist evolutionists is this is not an isolated thing. Human and sheep P2G pseudogenes. If you are familiar with the human sheep P2 pseudogenes of simply a gene that diverges after a speciation happened, share a coincidence, premature stop codon, which I mentioned a while ago, without the possibility of evolutionary ancestry. Okay, it is an independent formation that coincidence, which that coincidence premature stop codons. Okay, so there is no hierarchy around there. Okay, so don't be fooled. So I'm almost finished here. Sorry. Now the nested hierarchy associated here. If this premature stop codons occurred in sorry, I'm stammering again. If this premature stop codon occurred in a nested hierarchy, it would have been an evolutionary shared mistake. Okay, to be fair, which evolutionists are not. Evolutionists should discount all shared mistakes that have been inferred in the GC precursor. Okay, sorry, in the CG precursor, not just those that contradict evolutionary ideas. Okay, because they are biased. Sorry, again, the CG I refer to are the two of the GTAC of the DNA letters. Okay, GTAC DNA letters. Now, of course, the known natural behavior for some to many DNA sequences to mutate goes far beyond the CG. Okay, you following Standing for Truth, everybody? I wish I could simplify this, but we don't have enough time. Okay, it gets better. Nested hierarchies in the evolutionary literature has these specific numerical inconsistencies people seldom hear because they are hidden. So, or should I say, it is a trade secret? Okay, that's all. It is found all over their nested hierarchies. I'm just using the subcodons and the olfactory receptor here, and I can even use other parts of the body. Okay, so this is just like a nutshell for all their nested hierarchies of how I can disprove them by the numbers. Right. You disprove them standing for truth based on function, and I disprove them by using specific numbers. Okay, so there are more, but I will stop, let others speak. So that will be all, unless you guys need me to speak something else.
Well, what I like about that, Jeremy, is the olfactory receptors and the pseudogenes and the hierarchies that I guess they look to is one that we hear about a lot. Like I've read, especially in BioLogos, they look to that. So the fact that you're really taking your time to address that, represent their position accurately, and also refute it at the same time, I think is phenomenal. So
using numbers for the first time. Well, I never heard you use numbers, specific numbers in this hierarchies thing.
And I like it too because it's like a double whammy. We can look to function. We can look to specific numbers. For example, Ram and myself oftentimes will look at that highly conserved CO1 mitochondrial gene, and we'll look at the specific number of nucleotides, and we'll show gaps, and we'll show also the low diversity in that gene. That suggests that over 90% of, so we do oftentimes look to the numbers, the individual nucleotide differences as well as the function. So you're right, it's a double whammy. I love it. And
but I can give you the numbers of every single part of the body, from head to toe.
Right.
Over the hierarchies, even on other animals. I have collected those numbers in my archives. I'll try to look for it, and then we'll make another video, and we will slowly, one by one, refute their nested hierarchies using very technical numbers, very technical data, oxidase like that, and so on. So sorry, you guys.
No, don't be sorry. And that's why I would challenge too, in a respectful way, any evolutionist to object to what we're saying. And I know, even for the audience, I know a lot of what we've been discussing today, and I gave that warning at the beginning that it is highly technical, but at the same time, this is their proclaimed best and most technical line of evidence for universal descent. So I believe it deserves the attention. And, we talked a lot about protein moonlighting earlier. We went over just some of the literature, even though there really is massive amounts of literature that gives us, at times, unexpected functions of RNA. The fact that we functionally tested based on genetic knockout tests very, very little of the genetic language. We understand very little of the genetic language. Jeremy, you and I always talk about that. I like giving the analogy. It's kind of like if I were to go to Russia, and Jeremy, you used this before too, and I can't speak any Russian, and I went over to Russia to be a proofreader, and I looked at a book of 200 pages and I had to proofread it. I'm not going to know what's a mistake. I'm not going to know, just like when we're looking at the genetics, the pseudogenes, for example, they'll say is, these are shared genetic mistakes. But that's analogous to me trying to point out as a proofreader in Russia, even though I can't speak any Russian, what is a mistake and what is not. That's how little we know of the DNA language. And the fact that we predict that the vast majority of these DNA differences are functional, and evolutionists assume that they're all the result of mutations over time, that notion that the evolutionists have is slowly being overturned. Classic paradigms are being overturned. And that's why Herman Mayes, and just to represent their position fairly, Mayes will often look to, like, he's had numerous debates with Hovind, Jensen, and Jackson, and he used the same argument in every single debate. He'll look to a large number of gene trees, and he'll say that they all corroborate with each other. He'll point out that in phylogenetics, the best thing to look at is neutral variation. For the audience sake, that's just variation in DNA.
I don't agree.
Pardon, Jeremy?
I don't agree that we should look at neutral variations. We should look at the numbers that I presented.
No, I know. I'm just saying that this is what he says. I'm representing what he says.
He's trying to fool everybody.
And I rewatched his debate with Hovind earlier, Kent Hovind, so I know this is exactly what he said. And I rewatched his with Jensen and Jackson recently too, so I'm aware of his arguments. But you're right. Yeah, he'll say that that's the best. Conspiracy Cats, who I debated too, he'll say that the hierarchies in the cytochrome C protein, that's the best. So they've all got their own best, but it doesn't matter because the bigger picture is nested hierarchies, right? Herman Mayes is looking at neutral variation in regards to third position, codon variation. Conspiracy Cats is looking at mitochondrial DNA proteins. Regardless, it's all hierarchies is what they're looking to. So Mayes specifically, he's looking to that type of variation in our DNA, and this applies universally across species, that doesn't vary with function. Now, if the audience has been listening, right off the bat, we know that the evolutionist assumes lots of variation in DNA is neutral and non-functional to begin with. We don't make that assumption. So Mayes, his type of neutral variation, as I said, is the third position codon variation, for example. So to make it simple, we go from DNA to RNA to a polypeptide, and each amino acid, okay, it's encoded by a set of 3 bases. That's what we call a codon. We know that there are 20 amino acids that are encoded by 64 codons. Now, I Googled this just to make sure, just so I'm not guessing. I Googled valine, okay? We could have up to 4, GUG, GUA, GUC, and GUU will all make valine. All 4 of these codons will make valine. This means the third position can change and yet have no effect on phenotype, because the amino acid it's actually encoding is exactly the same. That's his point, okay? So these types, among others, is what somebody like Herman Mays would say are the best types of variation to look at when producing phylogenetic trees, because he's saying if there was a common ancestor, then these neutral changes must be passed on from those ancestors. Selection's not acting upon these neutral variations, because neutral means it's neutral to selection. Okay, so he'll point out that this observation on neutral variation cannot be true if independent ancestry is true. But the fact is, far from being neutral junk or just evolutionary leftovers over deep time, these so-called, degenerate synonymous codons actually and amazingly represent an independent secondary code in DNA. So even this paradigm is being overturned, because they're saying that there's a lot of redundancy in DNA coding for protein. We went over the fact that redundancy is good. It's good in computer code. It's good in it's good in the DNA code. But now we know that although statistically, because overall this is statistical hierarchies is what Mays looks to. It can be variable, but we're finding, and I've got papers here in front of me that show this, we're finding that it actually plays an additional functional role. They're not just redundant elements. They're not just tolerating error. The cell is actually using different redundant elements to slow and speed the process. It's information compression into every single letter, and it really is fascinating. So although these multiple codons, as I went over, may code for the same amino acid, the codons themselves have other functions. And the specific codon you're looking at is important, oftentimes specific to the kind of creature you're looking at. So once again, for the audience, boom, we both expect nested hierarchies. That is the rule. But what can differentiate between those hierarchies? Well, as Jeremy pointed out, we can look at the single nucleotide differences oftentimes, and we can look for gaps, which we've done. We can do that with the CO1 gene. And function. Function's key. But most importantly, what model is making the best testable predictions? What hypotheses has the best testable predictions that flow from them? And it is the creationists that are making the testable predictions on third position codon function. Dr. Jensen has like a 30, 40 page paper outlining very specific functional predictions on cytochrome C, mitochondrial DNA differences, third position codon functional differences as well. So genetics is where we win. And I believe at this point, genetics is where we are winning. So I'm pretty happy to say I think we've addressed every single one of their best lines of evidence for hierarchies. If you do have one last thing to say, Jeremy, to add to that, to be honest with you, I could go on for another hour, but we've been at it for 2.5 hours. I want to say,
like I said, I can go to other parts of the body and refute every single nested hierarchy.
I agree.
Historical values, but I want to give my time to gusto ko bigay sa iyo yung oras ko, Mr. Salvador Cordova.
Well, like you said, we can go on and on. I mean, like you said,
ikaw na mag-closing statement or kung ano man gusto mo sabihin.
For the audience sake, what we're looking at.
So I just said thank you. I actually didn't understand most of what Jeremy said.
No problem.
I'm just trying to fool my friends standing for truth here.
Neither did I. But for the audience sake, I just want to point out the fact what we're looking at here is a multi-dimensional genome. We're looking at, in regards to the third position codons that they say are degenerate, we're looking at multi-dimensional codes. And it's new levels of specified complexity. We see the same in the proteins. And we can predict that those mitochondrial DNA proteins are the same. So if there's, I know we went very technical. If there's anything, you know, I do want to end with this because from one of the studies, the authors said this, and I wouldn't be able to say it better myself. So I'm just going to read it. The authors in this, it's a secular paper on this, on the functionality of these third position codons. The author said, why the term degeneracy is completely inappropriate. This dual coding functionality of redundancy is anything but degenerate. It represents instead far more sophistication, layers, and dimensions of formal prescription. So remember, evolutionists are the ones assuming neutral, junk, oftentimes evolutionary leftovers. We're not, and that's why we have the prediction. So, yeah, Sal, thanks for joining us, brother. If you want, you can have as much time as you want and give your take on hierarchies. I know we talked about a lot there, but yeah, what are your thoughts, Sal?
Even though I'm a young Earth creationist, I'm considerably more cautious in my declarations. That's one reason I tend to argue from the what I call pedagogical creation models. Which means I'll argue for ID plus common descent or progressive creation rather than young Earth creation. And it's not because I believe in those models. It highlights the problems with evolution, and they don't get to. It prevents them from defining the debate in terms where I feel the young Earth creation model is not quite as strong as I'd personally feel comfortable defending. So I usually try to defend my models based on textbook biology, not necessarily young Earth creation papers. So, I mean, no offense to...
well, Sal,
yeah.
I just want to object to that because all of the papers where we go over like protein moonlighting, third position, codon redundancy, the functions found in there, the multiple functions, ERVs, ALUs, like these are all papers from secular journals.
Yeah.
You know what I mean? So we're not really using, is that what you meant? Like we're getting these?
No, what I meant is, I mean, just consider the people that are on our side of the debate by and large. Would be Michael Behe and Stephen Meyer and even Michael Denton. They're just killing evolutionary biologists, and they don't. Michael Behe believes nominally, not fully. He's nominally believing in common descent. He's open to changing his mind.
Right.
And his arguments just kill the evolutionists. I'm just like, well, where I get that from is. In mathematics, we have something called the proof by contradiction, where you actually assume the position you're trying to refute. And there's some very famous proofs in mathematics, like the square root of 2 being irrational. So I'm happy to grant, for argument's sake and argument for contradiction's sake, a certain model, because then because when we start talking about nested hierarchies, that's really their strong suit. It's their last defense.
Right. It's why we spent 3 hours refuting it. So,
I like going after their first. I mean, I don't even. I don't debate human evolution very much.
Yeah.
I attack it right at the root, the eukaryotic evolution. Then the next step would be something like chromatin evolution. I mean, that would be obviously before eukaryotic evolution. So let's say chromatin evolution, then eukaryotic evolution, then evolution of meiosis, then evolution of plants, then evolution of animals, and then nerve cells. You'll just kill them on each point. I mean, I prefer talking about that because I can show all these pretty pictures.
Right.
So I just wanted to say that's just kind of my personal preference. You talked about polyploidy. I only learned this recently. Humans actually have polyploidy in some of their somatic cells. And so there is function for polyploidy even in a diploid organism like a human. Our cardiomyocytes have like tetraploidy octo and whatever all the numbers are. They're like multiples of 4, especially in various stages of development. There are other cells in human somatic cells that have polyploidy. So let's, the evolutionists are really too quick to say that's just junk.
Right.
The biologists, the medical researchers who are studying human disease are very interested in finding the function. So as always, the evolutionists are really no help. They're actually just a hindrance. Wish they'd go away.
Amen.
They don't add anything. They add 0.
Well, the
So-
fact that they assume all this junk does hinder scientific advancement, because they're, we're the ones expecting function, and we're the ones that can actually, our basic presupposition can help advance science, especially in the medical field. But their basic assumption that the vast majority of the genetics is not doing anything, it's just based on genomic fossils, evolutionary leftovers, that you're right, that does hinder scientific advancement.
So when they throw that polyploidy thing, I'll just say, I don't know for now, but are you going to be really quick to say the polyploidy in human somatic cells is junk?
Right.
I doubt anyone would want.
Good point.
No one would dare say that. I mean, a medical researcher would probably be upset if you said you're not supposed to research that because it's junk. I can tell you there are papers on cardiomyocytes, and they think it's important for stress response, and they're very interesting papers on that. So I'm just saying, you guys, you evolutionary biologists have been saying this is junk. You said that about ALUs. You said that about transposons. I mean, how wrong can you guys get? I mean, how many times do you have to fail? How many times do you have to fail before you just give up? Because you're not doing anything. You're hindering science. And I'll tell you what junk is. Junk is 99% of what they publish. That's junk.
Amen. Good point. Yeah. And you know what? They are arrogant enough to put a lot of that junk in the textbook when they should be putting that right into the trash. And I like exactly what you said there, Sal. It's a really good point, because I'll often point out the exact same thing, because I'm sure you've heard it a million times when it comes to these functions, like of these mitochondrial proteins that are conserved. We all agree, evolutionists and creationists alike, that the classic biochemical functions of these proteins are very well known. Yeah. And they'll point to that and the hierarchies that they form as evidence for universal common descent. And we'll say, but that these sequence differences among these mitochondrial proteins are actually not also metabolically optimized or involved in cell optimization. Are you willing to put that on paper that's not the case? And they're not. That's the thing, because just like you said with the polyploidy, are they willing to put on paper that those really aren't functional, because they already screwed that up with the ERVs, ALUs.
I'll make a prediction, just a general one. Evolutionary biologists will find ways to make fools of themselves. That's just.
For some reason, our stream isn't working, guys. I don't know what's going on with our stream.
Oh, well, are we still recording?
I don't know. It's all black.
I just saw. Oh, yeah, it died on mine.
Yeah, it's still recording. But I have one question.
How long? Oh, yeah, it is all black.
Oh no! Do you want to restart?
Oh no, we can still hear it. I can hear you.
Salvador Cordova, may I ask one question?
I can hear the audio. I just don't see the pictures. Praise, is that your end, brother?
I don't know. I'm trying to fix it.
Okay, I hear audio on the monitor. Yeah, I just heard my voice.
Yeah, I heard your voice.
I have a second computer. I can hear it.
So I heard your prediction, Sal. I'm glad your prediction got on there, because that. That was a good one.
No, this is still. They could still hear us. So, guys, who are watching, thank you for listening. And Jeremy had a question you wanted to ask me.
Yeah, just quick answer, quick Q&A. This is something I don't know. I am a person who is atheist. I consider myself atheist. I respect Dr. Francis Collins and Dr. Hugh Ross. They are both. Dr. Francis Collins is a evolutionary creationist, and Dr. Hugh Ross is an old Earth creationist. I learned a lot from